kind enough to join us from Sydney, fairly early in the morning, so much appreciated. Um, he has a really interesting research program. He has published quite a lot of very interesting work on Parkinson's disease, particularly using functional imaging methods, but has also published quite a bit on very interesting work, sort of more basic work, looking at basic organization of brain circuits and brain networks. And I think that's really the perspective that's informed a lot of his work. And so that's what he's presumably going to talk about, mainly his PD work today. But against the background, I think, of a lot of this very interesting sort of higher level circuit analysis and discussion. So again, thanks so much for joining us, and please go ahead.
Yeah, thanks very much, Roger, and a real pleasure to be here. Kind of went back down memory lane a little bit when preparing this talk and thinking a little bit about the sort of journey that I've been on academically, kind of diving into the complex and challenging world of trying to understand Parkinson's disease and its kind of inherent complexity. And then, as you say, sort of traveling then from the Parkinson's brain into the kind of world of healthy brain function and just trying to make sense of this complicated sort of knot of neuroanatomy functionally. So I'm actually going to talk a little bit more today about some of the more recent work we've done, but I'm going to do it in light of kind of where I see, or at least I saw when I kind of was more invested in the Parkinson's world, where I saw some of the really interesting curiosities and challenges in linking models to symptoms. All right, so I'll just share my screen now. Can everyone see that okay? Get a thumbs up, maybe. That's okay, Roger?
I think you're in the, yeah, there you go. Okay, got it. Okay, you're set there. Great.
Okay, great. All right. So, as Roger mentioned, I'm at the University of Sydney on the underside of the world. It's actually tomorrow for me right now, for you all, Thursday morning, about 7 a.m., so not too early. And I was introduced this morning to Roger by the knowledge that my previous PhD supervisor actually got up earlier than me. So there's nothing like a little bit of intimidation early in the morning to get you started for a talk. All right, so what I want to talk to you about today, as Roger mentioned, my group is really interested in this kind of big, nebulous, complex problem of trying to make sense of the brain, the organization of the nervous system across scales, trying to think about the cellular level, but also the level of macro scale where we image the brain, and to try to bring that to bear to understand some complex behaviors. And so I love to show this video at the start of talks, just to remind people just how far away we are from understanding what a nervous system can do. This little child here, probably no more than 10, is balanced on this little board, wibbling back and forth. His eyes are locked on a target in the distance. He doesn't deviate from them at all, like a hawk stalking its prey in the breeze. He's got this wind behind him. He's got his hockey stick flipping this puck that he never even looks at once. And I'm not a particularly big hockey fan, but I just think this is a remarkable display of really competent parallel processing, flexible behavior, goal-directed behavior. And we don't really, I would like to argue, have really great explanations for how on earth it is this child can do this function. We have areas of the brain we could imagine being impaired that he would struggle to do this function. We might say something like, oh, if this gentleman had a lesion in his cerebellum, he might not be able to balance on the board. Or if he had a lesion in his basal ganglia somewhere, he might not be able to parallel process and do multiple things at once. But I don't think we have good functional explanations for how that normal system works in the case of this healthy young man. Now, obviously, for most people on the call, the target that you're trying to hit, or that you're trying to make a difference for, is people with Parkinson's. And I think we actually, so when we think about Parkinson's, a lot of what we could think of is almost the sort of lack of the ability to do a lot of what the boy there could do on his little balance board. Obviously, the boy had a bit of a blank face facial expression because he was focused, but he probably could have been expressive if he wanted to. He had really flexible extremities. He wasn't shaking at all. He was quite focused with his movements, and they were quite goal-directed without any extra added amplitude to any of the movements. His posture was balanced. He wasn't tilted forward. His arms were swinging around as he wished. They weren't reduced, and he obviously wasn't walking, but you could imagine that he would walk well as well. And so, obviously, there's a long and storied history trying to understand what's gone wrong in the brain of Parkinson's disease. And we all know, I'm not going to belabor point, but we all know that one of the really key early discoveries was this lack of dopamine in the substantia nigra, pars compacta, projecting up into the striatum. And so from there, we could take our kind of models of the cortico-cerebellar-basal ganglia thalamic system. We could kind of wire them up in a way that we think marries up to the neuroanatomy. So we know that there's strong cerebral loops between the thalamus and the cortex. We know there's particular pathways connecting the cortex to the striatum, which then gate inhibition in the globus pallidus. That has inhibitory control over both ascending systems in the thalamus, but also really complex descending systems in the midbrain, pons, spinal cord, etc. And we also know there's these interesting pathways that connect between the cortex and areas of the extended hypothalamus, like the subthalamic nucleus, that then project in and drive glutamatergically the output structures of the basal ganglia, as well as modulate in a complex fashion areas like the cerebellum. And so in Parkinson's disease, when we lose that neurochemical dopamine, we essentially are changing the excitatory milieu of particularly the striatum, but not exclusively so, and I'll discuss that a bit later in my talk. And the flow-on effects then of that lesion are to create something like an information lesion in the system. You essentially lose the gating over the globus pallidus. This now has this increased inhibitory control over the ascending and the descending systems, and you sort of render yourself open to these the firing of the subthalamic nucleus, which otherwise was gated a little bit by the inhibitory control of the striatum. And so if we think of a lot of the symptoms in Parkinson's disease, the ones that I mentioned before, I think they're actually pretty satisfactorily explained by this idea of a sort of relatively global inhibition. You can imagine it fluctuating over the course of the day in context, but really relatively global inhibition over the mesocephalic locomotor region and its surrounds, central pattern generators and the like. And the idea being that with an increased inhibition over the kinds of brainstem structures that would normally automatically control your posture, your gait, your ability to maintain truncal tone, that you would essentially lose the ability to make those flexible and adaptive. They'd become rigid. And I think that's quite sensible. But as we also all know, many other symptoms characterize the Parkinsonian syndrome. Depression, cognitive impairment, hallucinations, freezing. These are just a few that I mentioned, in part because I spent a lot of time in my PhD, as Roger mentioned at the start, performing functional imaging on individuals with often akinetic rigid Parkinson's disease and self-reported visual hallucinations and freezing of gait, and really trying to understand these other much more nebulous and complex multi-system symptoms that also have another very key feature that I don't think is as well explained by this notion of a sort of tonic inhibition of the brainstem, which is that they're relatively paroxysmal. And what I mean by that is that they're not always
Hey
expressed.
Noah,
So,
why don't we scan the river meat stuff?
Oh,
oh,
hold on.
someone there.
Somebody needs to mute themselves.
It's a great question. I have no idea, Noah, what the answer to that one is. Yes, we have this. I think we can characterize the symptoms of Parkinson's very, very loosely here. I'm not trying to use any kind of objective criteria here, but on one hand, on the left, we're going to put symptoms that are relatively pervasive, the kinds that are relatively consistent when you have a person, let's say, with Hoehn-Yahr 2.5 Parkinson's. Their arms aren't going to swing very well. Maybe they'll get a little bit of a benefit from a bolus of dopamine, but that'll wear off, and they'll maybe start to get symptoms from having too much dopamine around. But I put this other group of symptoms in a different category, which is that they're not always expressed. So someone with freezing of gait, they might be able to walk through a doorway a few times, but just on that fourth time, their feet stick. Or you get them to perform serial sevens, and they're quite fine for the first 2 or 3 trials, but on the fourth trial, then they become impaired and they have a problem with their gait. And these kinds of symptoms pop in and out. They're not always pervasively present, rather they're paroxysmal. And as I mentioned, I don't think that thinking about how the brainstem is inhibited can really help us here, because oftentimes what we're talking about in these paroxysmal symptoms is a much more complex multi-domain trigger. So in the case of visual hallucinations, it might be something to do with the sleep-wake cycle, and it might be to do with the complexity of the surrounding environment that the person sits in. Are they in a familiar environment versus a novel environment? Are they challenged by someone around them, or are they really focused? And these kinds of features are a little bit harder to explain with the brainstem, but they actually make a lot of sense, I think, and what I'd like to try to argue today, when you start to take very seriously the notion or the anatomy of the thalamus. And so what I'm going to do today is essentially speak about the thalamus and some of the work we've been doing there, and then try to essentially motivate it to bring back in more nuanced understandings of the architecture of the basal ganglia. So when I was approximately 2 years old, this absolute gangbusters of a paper that I think has just really dominated the field was laid out for us, and I think going back and rereading it for this talk, I was again struck by just how visionary this team was. It's very sad to hear as a young neuroscientist of the passing of Marvin DeLong, and I think he left just a gigantic legacy behind him. And I never got to meet the man, but I heard just lovely things from people as well that got to meet and work with him. So this idea of parallel loop architectures connecting cortical regions through the striatum, globus pallidus, back through the thalamus to the cortex, I think is really the kind of the leading theory about how the basal ganglia system is organized. But one of the things that really struck me when I went back and read the paper again for this talk was just how cautious and careful the authors were to essentially both advocate for the confirmation of the anatomical principles with modern techniques, and also open to the notion that we might advance the model once we learn more. Particularly with respect to, their argument was for the pallidal connections to the thalamus. So this last loop here, they said that we really knew nothing about that. But what I'm more really interested in is what these connections from the thalamus back to the cortex might add. So just to kind of motivate that, there's this really interesting challenge to this parallel loop model, and I actually mentioned this in the original paper as well, which is that the system itself is sort of in many ways not organized like a set of parallel loops. It's organized more like a funnel. And again, people have known this for a very, very long time. But one of my favorite papers when I was a grad student was from Charlie Wilson at UTSA, where he argues for this sort of information reduction notion, where you've got a lot of cortical neurons, let's say multiple millions of neurons, projecting down to a couple million striatal neurons, which then collapse further into a couple 100,000 pallidal neurons before getting to, he doesn't draw the thalamus here, but again, some thousands of thalamic neurons before projecting back out to the cerebral cortex. And so you immediately notice that it's quite hard to imagine how parallel loops could essentially form when you're essentially losing some of the information at each of these steps. You're reducing the dimensionality. So really from an early age in my foray into Parkinson's, I was really curious how it is you could accommodate these two different perspectives. It's really akin like this cat's seemingly miraculous ability to get out of this tiny gap. You somehow having to squeeze all of the synaptic information through this circuit before it comes back to the cortex. And so this is actually a drawing from the original paper where they actually had a cartoon of what this might look like. You've sort of got some cortical neurons projecting down to a few neurons in the striatum, collapsing even further into the pallidum, and then back to this kind of magical combination ABC neuron in the thalamus before coming back to the cortex. But so what I really want to drill in on today is this link in red. What do we know about the projection of the thalamus back to the cortex, particularly the pallidal receiving region? And what can we say about it computationally? And then if we take that seriously, what might that help us understand about the Parkinsonian syndrome? So what I'll do in my talk today is I'll walk through just a really brief overview of the thalamus itself. I'm not going to be able to cover it in a great amount of detail, but I'm more than happy to answer questions afterwards. I'm going to particularly click on a subtype of the thalamus, subtype neuron of the thalamus called a matrix type or a diffusely projecting type. It's a calbindin staining neuron that actually projects to multiple different regions of the cerebral cortex and hits often supragranular or layer 1 of the cerebral cortex in a really different way than the traditional LGN type loops that we learn about in medical school. I'll then talk to you a little bit about some of the modeling that we've done to try to understand this neuron, this matrix type neuron, using a technique called neural mass modeling. Again, I won't belabor a lot of the details, but I'd be more than happy to double click on them. I'll then talk about some of the subcortical inputs to the thalamic system and how they relate to this architecture. And then finally, if I have time, I'll talk a little bit about how neuromodulation affects this distributed circuit. All right, so And please feel free to interrupt at any point if there's anything you'd like me to double click on or unpack. So this is a little cartoon of the thalamus from a recent review that I worked on with a few colleagues. So here in purple is the anterior nucleus, all the way back to in yellow the pulvinar. This little green P thing at the bottom is the LGN. They're actually a bilateral structure, and so you're sort of sitting in the midline. And I think they're actually quite a fascinating structure because they're nuclear, like the hypothalamus, where they've got multiple different subnuclei packed into the same anatomical volume. And that's quite distinct from something like the striatum, where you've got this kind of spread or spectrum of gradient of connectivity across the anatomical system. So the interesting thing about these different nuclei is they project to very different and quite particular parts of the central nervous system, but also receive inputs from each of them as well. So the medial dorsal nucleus in green will receive very different inputs and project to different areas than the anterior nucleus in ventral anterior ventrolateral. So it's not a homogeneous structure. It's quite heterogeneous, but really, really low dimensional, and also still a coherent whole, which I'll argue for a little bit later. If we zoom out and think about the connectivity of the thalamus, particularly the ventral tier, so here I'm focusing more on VA, VL, and MD. They have this really interesting relationship with the basal ganglia and the cerebellum, which I'll click on later. But the point I wanted to make with the diagram here is that sometimes when we think about the thalamus, we think about it just with respect to the cortex. But it's very important to remember that its architecture and the nuclei within it have actually explicit rules for their connectivity with areas like the basal ganglia, particularly the globus pallidus, other parts of the extended inhibitory system like the zona incerta, the pretectal nuclei, but also areas of the cerebellum like the deep cerebellar nuclei. And I think that if we take that logic seriously, then explanations of higher brain function can start to make a lot more sense. So as I mentioned, so here are all our little nuclei here. And if we were to zoom into the microscopic level, we'd actually see that there's two really different types of projections that come out of the thalamus. So there are a lot of interneurons as well, which I won't focus on today. They're GABAergic and locally projecting, but the long range projecting neurons that come out of the thalamus can be kind of classified into two main types. So this is an idea that's been around for some time. Ted Jones, who literally wrote the book on the thalamus, was a big proponent of this idea. It went out of fashion for a little while when people got really interested in the notion of tracking what were called first order and higher order thalamic nuclei. And the idea there was trying to determine whether or not the thalamic nuclei was receiving an input from the subcortex as its driver or receiving an input from the cortex as its main driver type input. And if you characterize them that way, you can kind of differentiate all the different nuclei within the thalamus and sort of label them as different types. Ted Jones had a different take, which is let's look at the thalamic nuclei themselves and ask whether or not the majority of neurons within each of these different subdomains projects to the cerebral cortex in one way or the other. And the two different ways he identified, one was this core type, which projects in a really targeted way to the granular layers of the cortex. This is layer 4. And so the main LGN pathway that we learn about in med school, the retina projecting into the LGN, projecting on to layer 4. This is the kind of main prototypical type of that pathway. But interdigitated with those nuclei, those neurons in exactly the same nuclei are this completely different, diffusely projecting what he called matrix type. And if you look down the microscope, it looks like there's a sort of matrix of these blue types in the background with little pockets of the red type as a core on top of them. That's where the name comes from. But importantly, they project up to the supragranular layers of the cortex in a completely different fashion than this core type, and they actually often spread over multiple different cytoarchitectonic regional areas. So that's obviously a simplification. There's many different subtypes here. I don't want to give you the impression that there's these two different types and it's sort of A or B. In fact, there's a panoply of these weird things spreading all the way from the core type in here in blue in this slide to this more matrix type in pink. And there's even a type that you may have heard of called an intralaminar type. This is a really fascinating cell type that actually projects more exclusively to the striatum than it does to the cerebral cortex, and it has sort of weak projections to the cerebral cortex. Lots of the amygdala looks like this as well, the central amygdala. But the point that I wanted to make with this slide is that these different cell types, if we look over here on the right, we're now looking at different thalamic nuclei, are actually sort of spread across the different nuclei in ways that don't always respect the psychoautotectonic background. So you can see that in the ventrolateral nucleus, you see a lot of the core type. These are actually the ones that receive deep cerebellar nuclear output, but you also see a bunch of the matrix types, both of the diffuse and the focal. So there's a big spread of these different types across the thalamus. They're relatively local, these matrix type, which I think is kind of a fascinating result that's been out in literature just recently. So even though they do project up here to these multiple different areas, if you were to go through and put a adenovirus directly into the parts of the thalamus here, so the ventromedial thalamus, and you then track the targets of those different viruses as they spread throughout the nervous system, they essentially run along the axons of the area that you've infected. You can actually see that they infect slightly different areas of the cortex. So here in the ventromedial thalamus, you've actually got areas that are projecting to parts of the motor cortex in yellow and blue, and then parts of the anterior cingulate in pink, and they don't really overlap very much. But it's important to note that even though they do spread to different areas, within those areas, they actually spread over multiple different, what we would call functional boundaries, in the sense that in that area, there's going to be multiple different cortical columns, and these cells will actually project to multiple different cortical columns. So they're doing something relatively diffuse, but with an interesting spatial topography as well. Importantly for our discussion today, it's actually been known for some time, and this was again one of my favorite papers as a grad student, 7 problems with the basal ganglia from Nambu. It's been known for some time that the different types of thalamic neurons that I mentioned, particularly in the VA-VL complex, which we know supports the motor cortex and pre-supplementary motor area and supplementary motor area, that the cerebellum inputs and the palatal inputs actually have a predisposition towards these 2 different types. And it's actually kind of counterintuitive, because it's not really the way that you might imagine from if we drew parallel loops around the system. We might imagine that the basal ganglia was gating these targeted types so that any cortical message that was passed down through the basal ganglia could hit this targeted type and then allow it to come back and create a little resonant loop in the system. In contrast, it's actually targeting these matrix types. It's targeting the types of neurons that come up and then spread up into these supragranular regions of the cortex and diffusely kind of innovate this sort of canopy of the neurons in this in the supragranular regions of the cortex. And to me, this is actually deeply fascinating mystery, because the Alexander DeLong and Strict model is incredibly predictive, has been extremely helpful for, I think, the field understanding the symptoms of Parkinson's disease. But then here's this anatomical feature that doesn't fit with our first order intuition as to how that system might be wired. So I think it's still working most probably the way that they suggested. Maybe the mechanism by which it's working is a little bit different than we anticipated. And maybe through that difference, we can develop a better understanding of what might be going wrong in the brain when someone develops Parkinson's disease. This is just to show you some modern evidence from a really lovely paper by Adam Hampman's group, showing when they put in retrobeads to different parts of into the thalamus, as well as into the deep cerebellar nuclei and the substantia nigra. You can see the different inputs into so from the substantia nigra into VA, and then the deep cerebellar nuclei into VL. And if you then looked at the different types they actually then tracked each of these neurons around with more modern techniques to look at the proportion that we're actually projecting to layer 1 in the cerebral cortex, the kind of most outer layer. And I've just highlighted here the basal ganglia targeting neurons in little red circles, because their graph isn't really designed to show you this. But they really have something a lot like this red type here. The projections are coming up and hitting areas just in the boundary between layer 2, 3, and layer 4, and then layer 1. So then the question becomes, well, what's so special about layer 1? Why would we be interested in what's happening there? And so this is a really fascinating area. In fact, Ramon y Cajal, one of the great grandfathers of neuroscience, actually called this the crowning mystery of the cerebral cortex in some of his writing. And he obviously had a poetic edge to him, as well as a beautiful eye for art, because he was quite a great illustrator of the nervous system. But what's really fascinating about this area is that it's really, really scantily populated by cell bodies. And so if you were to look at the cerebral cortex, so here we can imagine the peel surface, a bit of skull, there's the pia here. If we went down deep into the and we hit the white matter, we're essentially going through the different layers. And obviously there's going to be different cell types that whose cell bodies live in these different layers. And we often name the cells according to where their cell body lives. So we'll call a cell a layer 4 stellate cell, or a layer 2-3 intratelocephalic cell, or a layer 5B pyramidal neuron. But you can also notice from this slide as well that a lot of times these cell bodies actually aren't really that descriptive about what this cell's capacity might be within the circuit. And you notice that in yellow, and you can't see it as well in the orange, but I'll mention them more in detail in a second, that a lot of these neurons actually have really, really long apical stalks that project up into the supragranular layers of the cortex. In fact, I don't know how many of you can see my picture of my video, but this beautiful drawing behind me from Greg Dunn is actually a drawing of these layer 5 pyramidal neurons with their projections up in the apical stalks up into layer 1. But as I mentioned, there aren't really any cell bodies there. But there's a lot of white matter tracks. And so a lot of the white matter tracks that come from higher order cortex, so more agranular cortex, this would be cortex that's towards the area of like the anterior cingulate and frontal areas, are actually projecting back towards more posterior areas and landing in these supragranular layers, as are a lot of those matrix thalamic inputs. And so I think that intersection between these really, really important cells that have their cell bodies deep with their stalks up in layer 1, and the intersection between those feedback inputs and the intersection between the thalamus is a really critical nexus for thinking about whatever kind of information processing is being conducted by the cerebral cortex. So why is this cell in particular one that we're really interested in? Well, we came across a few years ago some just absolutely beautiful work from Matthew Larkham. He's an expat Aussie, but he lives in Berlin now and works at Humboldt University. And Jan is a theoretical neuroscientist who works in Estonia. And they've been working on what I think is a really beautiful idea that helps to provide a real biological grounding to ideas of predictive processing or predictive coding that are, I think, a really great algorithmic explanation for how our brains can essentially process information. And the reason I really love it is that it really puts the neuroanatomy to work. It forces the system to connect at the cellular level, that I think is really, really nice grounding from which we can build a lot of our understandings of how the nervous system works. So the system as a whole is based on really painstaking patch clamping that was done on this neuron type. What that means is that they take this neuron in a dish, they go in and they put a little pipette onto the surface of the neuron, but they don't disturb it. They basically are listening in like a wiretap on that neuron, and they can go in and they can project in different types of neurochemistry on each part of the system, each part of the cell, and they can actually understand what happens when they perturb this part with current and that part with current, what happens if they block this neurotransmitter, etc. So they can understand in exquisite detail. And it turns out that this cell type has a really, really interesting phenomenon, which is that because it has such a huge long stalk, it looks almost like a palm tree, the kinds of inputs that it receives to the apical dendrites are actually really disturbed and distant from the inputs it receives to its basal dendrites, such that most of the time, any message that gets hit onto one of these dendrites tries to travel down, it gets lost on its way. It kind of leaks out of these different leak channels and currents that it has on its stalk. But you can close that gap with something like the thalamus, and all of a sudden you can cause this system to fire in a burst firing mode. It actually causes a calcium wave whenever you get an input here that propagates back up. If the thalamus is there, closes a channel, that means that the next input can essentially cause the cell to burst. And so why am I giving you all this kind of this detail? Well, the reason is that I think this is actually a really, really important mechanism for matching your experience or with what you expect to see. So the idea is that when, let's say, some visual input hits your retina, you pass some feed-forward sweep of input through your LGN to, let's say, the basal dendrites of these neurons in a circuit, which then sort of pass up some hierarchy of the kind of evidence that you've seen. But if you imagine a dog, you're going in the opposite direction. You're essentially priming the apical dendrites of these neurons so that they can essentially expect there to be an input. And when these two things match, when the expectation matches the evidence, you get burst firing in this circuit, and this neuron is now capable of actually enacting some change. It can cause movement if you're in the BET cells, or it can get your colliculi to shift if you're in the parietal lobe. And there's a weird and wonderful world of these different cells. They're essentially ubiquitous across all layers of the cortex. But Matthew's done some really, really painstaking work in rodent models, really confirming that these cells are really, really crucial, and the burst firing of these cells, particularly when these two compartments are triggered within the same relatively contextual window, is really crucial for memory formation, conscious perception, motor action, and many things in between. And so we're really interested in asking, okay, what about this idea of the thalamus and the matrix thalamus gating the coupling between these different cell types? And the reason it's, I think, so interesting to us is that the thalamus is organized in such a different way in the cerebral cortex. As I mentioned before, it has a really low dimensionality, and we're really interested in understanding what that constraint could impose if it gates multiple of these different pyramidal neurons at the same time and allows them to enter into the burst, but not these ones to enter into the bursting regime. What impact might that have on nervous system function? So yes, I've just undercut my point. So the cerebral cortex is really, really different from the thalamus in that it has just far less neurons, and this actually imposes one of those dimensionality constraints. I'm not going to belabor the point now. We've done a bit of neuroimaging that shows that if you take whole brain neuroimaging and essentially look for a low dimensional readout of neural activity, that the thalamus itself is really, really strongly corresponding to these low dimensional patterns that we can see. This is during a task a little bit like Sudoku. When you do an easy versus a medium versus a hard version of the task, you see this particular change in the pattern of these low dimensional signatures over time. And different parts of the thalamus, in particular the medial and the posterior compartments, are really mimicking the same time series we see. This little line here is just dotted on this graph. We're seeing really, really similar patterns in these two different compartments. And so we're really interested in this notion of essentially taking seriously the idea of the thalamus and its constraint on information processing in the rest of the system. So a way to make these ideas more explicit, rather than just telling you that the thalamus has less neurons than the cortex and therefore it has a constraint, we can actually build models that have these assumptions in them. So a really talented postdoc in my lab, Eli Muller, has a background in physics, statistical physics, and neural modeling. And so what Eli likes to do is to essentially listen to me bang on about my favorite bit of neuroanatomy, tell him about how beautiful the layer 5 pyramidal neuron is, or how cool the striatum is, or something, and then try to see if he could synthesize a really simple wiring diagram model of what that system might look like, and then test some of our different intuitions about how it works in ways that betray its function. And so in this case, what we were really interested in doing was taking an existing neural mass model of the corticothalamic system. So here, what we've done is we've essentially zoomed out and thought about, don't worry so much about these layer 5 pyramidal neurons. Let's just worry about a pyramidal neuron itself and its interneuron. And they're connected into a little loop with the core projecting type thalamic nuclei with a reticular nucleus inhibiting in a little loop. And then what we do is each one of the little circles in our little model is one of these little loops. And then what we do is we wire them up into a little toroid, a little donut. Physicists love this because you essentially can't fall off the edge of a donut. Everywhere you walk, there'll be another bit of the donut. And that's really nice because it means you don't have any periodic boundary conditions and your mathematics are much easier. But we don't really think the brain is a donut or shaped like a donut. It just makes the maths much easier. But what we wanted to do in this study was essentially just sweep a single parameter. So everything else stays exactly the same in the model. All we're doing is we're asking what happens if the model receives something like this matrix input, this diffusely projecting input to the whole system, and what if it gets none of it? And what we really want to know is what happens if you take a low dimensional pattern like the thalamus is projecting and give that access to multiple different regions that are mimicking what we think the cerebral cortex is doing. And it turns out there's actually a really beautiful intuition here for people that are uncomfortable with mathematical modeling, is that the brain actually ends up working an awful lot like what happens when you boil water on your stove. When you start heating up the system, the diffuse projection parameter is really just like what happens when you heat up the dish. So what you're really doing is you're increasing the kinetic energy of the system. Now more of the different water molecules can transition into to gas from liquid. This is a little bit like a brain region becoming active versus non-active. But you also increase the diversity of that state too. So there'll be little pockets that can bubble a little bit more than others. And that's just because when you start heating up your water, what you get is a bunch of different water molecules get bounced into by just in one little pocket of time, get bounced into by a few of their different water molecule colleagues. And there'll just be just as many that get ignored. And this is because we've increased the diversity of the state as well by increasing its kinetic energy. And what that means is that little pockets of your water actually think that it's hotter than it actually is, and they actually transition into a gas. But because the system as a whole isn't actually that hot, they can't stay as a gas. They have to fall back into liquid. And the reason this is so interesting, this kind of sort of bubbling regime, is that it's actually a really great way to have information processing without the entire system flipping over into this different state. So you imagine if I turned up the gas too high, if I had the temperature, let's say, a few hundred degrees higher, the entire system would burn off. There'd be nothing left. There'd be no ability for me to continue to allow some pockets to bubble in the future. Because if I had the temperature just right, what I can do, and I can turn it up and down, I can now make it such that certain pockets of this water can transition into gas, but then fall back down into water. And we think this is actually a really, really important feature of this diffusely coupling system, that it permits the multiple realizability of the activity of the system. So I know this is a concept that wouldn't typically come up in a neurology lecture, but to me, this is such a beautiful potential algorithm for what the diffuse system of the thalamus is potentially providing the cortex, that I really wanted to click on it for us here today. And I think it's actually really intuitive when you think about it from first principles. So I won't belabor the point about criticality. So the real point that I want to make with this slide here is that it's really not about being in one or the other regime. It's about being able to poise yourself in the middle. You want to be able to have a blend of states that have a relatively inhibited thalamus and a blend of states that have a relatively activated thalamus. In this case, the reticular nucleus is blanketing the relay nuclei. In this case, the relay nuclei are winning the battle and they can make it back out. And if you can blend these two different states, now all of a sudden you can process information in a really, really nuanced and rich way. Just to, in case anyone's interested in slightly different areas of neurology, there was a fascinating recent study by Yuri Solomon and colleagues in Wisconsin, where they anesthetized a macaque monkey with propofol, and they actually went in and stimulated part of the matrix thalamus. In fact, this case, it was called the centrolateral thalamus with electrical stimulation, and they could actually wake the animal up from anesthesia. And Eli was able to basically take that very similar model to the one I just showed you and recreate those effects. So we can essentially take stimulation of the brain and recreate neuroanatomical signatures that we'd get from neuroimaging. So these are the kinds of things that you can easily measure in the clinic. And while we have not pushed this into the domain of Parkinson's yet, I'm really, really interested in the idea of taking neural recordings from individuals with Parkinson's, whether they be in the theater, or if it's, people that are outside the scanner, outside the theater, in a scanner doing, an fMRI experiment or EEG, and trying to use models and manipulate parameters of the neurobiology in the models to recreate the kinds of signatures we see in the clinic, because I think they'll give us clues as to where to try to actually interrupt the system's abnormal dynamics. So if anyone's interested, please feel free to reach out later. All right. So I mentioned earlier on as well, so these we're arguing that these matrix thalamic nuclei, these blue projections here, are doing something like acting like a temperature parameter for the rest of the cortex. They're essentially allowing different parts of the cortex to become active and process information in that notion of expectation and evidence matching. And they're really reinforcing your ability to do that in the particular domain in which they're projecting to. But I also mentioned at the start as well that there's a curiosity here, which is that the basal ganglia outputs are actually projecting to this diffusely projecting type, when we often think about them in the sense of projecting to these core type. Now, I've argued in some more theoretical work that actually this makes an awful lot of sense when you think through the different architecture of these two different systems. And so just in cartoon form here, the basal ganglia, as I mentioned before, have this really interesting dimensionality collapse where the striatum collapses down to the globus pallidus, which then collapses down to the, so this would be the pallidus externus down to the pallidus internus, and then here back to the thalamus, which now projects back out to the rest of the brain. And so we've taken to really thinking about this system rather than being involved in action per se, are involved in the idea of being able to be flexibly active. So in other words, you can allow the system to function. You're allowed to activate the particular subset of neurons downstream, but you're also at the same time bringing along a whole bunch of other different potential neural columns that could help you to be flexible in that context. Was there a question there? I heard a noise.
So just one question. So let me, didn't have a question, but let me briefly interrupt. So with this attractive sort of phase change model that you have, but this looks like it's sort of a this takes us back to really old ideas, sort of a non-specific activation effect. So how do you get how do you sort of bake specificity into the information transfer?
Yeah, no, it's a great question. So I think one of the really curious things about this circuit is that the flexibility that you're buying is a flexibility in potential, but you still need the information streams, let's say the thing that you're hoping to achieve and the opportunity to achieve that to match together in order to get the system to burst. So these thalamic projections will not activate the neurons on their own. They'll just make it more likely that those neurons will activate when they're given an opportunity to do so. So you think about it more like a priming type of an effect, then you think it more like a message being passed that now's the time to act. And we think that that actually provides essentially more grist for the mill, that you've now got flexibility in your movements. If you were to lose that, you can imagine that the movement that you enact at the start is now locked in. There's nothing you can do about it. It's essentially stuck as the movement you have to do, and you've got no wiggle room around you. If someone moves the cup from my desk 2 inches to the side, I now can't shift my motor plan to actually grab the cup. It's now I'm stuck grabbing it the way that I was going to grab. You become essentially perseverative in that motor action. That's the prediction.
Okay.
All right, so I'll forge ahead and hopefully bring it back to the land where we can have some fruitful discussions, because I do I do appreciate that a lot of this is more theoretical neuroanatomy, but I think the reason that I'm so excited about a lot of this is that it's really grounded in cellular level anatomical detail, such that whatever extrapolations we can make, I hope will end up having some benefit for people with disorders like Parkinson's that struggle with these kinds of otherwise inexplicable phenomena that are really, really hard to understand using our traditional models. So I mentioned this idea of this sort of basal ganglia collapse back to spread. Just in cartoon form, the cerebellum is doing something completely different. So here, the main cells of the cerebral cortex that I've been mentioning project out to an area in the pons, which then projects to a gigantic population in the granule cells of the cerebellar cortex, which then hit Purkinje neurons. They collapse back down to deep cerebellar nuclei, which then project back out to the core nuclei of the thalamus and back to the cortex. And I don't really want to belabor a lot of the cellular level details there, because we could do it a whole hour on each of those on their own. But just to say that if you can have different cortical cells recruiting these two different populations of subcortical systems, the likelihood is that they're going to be able to do completely different things. And we've argued in this work that essentially that the cerebellum is really, really great for predicting the sensory consequence of the action that you're currently taking. And that's going to be really beneficial for things like anticipation, for being able to essentially take advantage of what the opportunity might be going forward. And essentially, if you turn off the thalamic inputs to the system, you're really shutting this down and trying to force this system to do all the work. And I think if you think about Parkinson's disease and a lot of those more complicated symptoms that way, that can be a really really interesting inroad into insights for these different symptoms. All right, I'll skip over these couple slides here. I wanted to say a last thing about neuromodulatory control, because I find this just deeply fascinating. And I think it's, again, a very helpful extra little detail to sprinkle on top before we come back to Parkinson's. And so we all know about the neuromodulatory system. We think a lot about the ventral tegmental area in Parkinson's disease. But I saw Nico Bonin was on this call. He really opened my eyes as a grad student to the notion that the cholinergic system and the noradrenergic system and the serotonergic system are also really, really important factors in trying to understand the various symptoms of Parkinson's disease, because they all in different time courses develop synucleinopathy and cell atrophy that can be potentially explanatory for these different symptoms. So while these nuclei are spread across the brainstem and the forebrain, they're really different in that they project completely different ways to the rest of the brain, and they release different types of neurochemicals. But they do have something in common, which is that they have a really common mechanism of action when they target cells. So rather than using ion channels, which let's say you release glutamate across the synaptic cleft and you hit an ion channel, you can then pass the glutamate through that channel, or you open that channel, sorry, and then sodium can rush in and you can cause an action potential. These neuromodulatory nuclei typically like to use something called a G protein coupled receptor. So these are now transmembrane receptors that have an internal domain. And whenever the ligand binds to this receptor, it causes a conformational change that then causes a second messenger cascade in the cell that either releases calcium from internal stores in the endoplasmic reticulum or opens up different ion channels like potassium leaks and calcium channels and of the like that can really change the internal milieu of the cell. And rather than causing a cell to fire an action potential like one of these ion channels, really what they're doing is they're changing what we call the gain of the cell. You can think about this a little bit like the volume knob on the cell, whether or not the cell is listening into any of the inputs that it receives or ignoring them. And another way to say it is these neuromodulators are changing how much bang you get for your buck. There's going to be a signal coming in. How much do I translate that into an output? So Jim Surmeyer had this really lovely idea that I think is really, really helpful when thinking about the effect of dopamine in particular on the circuit. And I want to extend that a little bit today to incorporate some of these ideas that we've been talking about. So the idea being that when you're at rest and there's no dopamine, let's say the dopamine levels have dropped, D2 cells are actually much more active. These would be the indirect pathway cells that project to the globus pallidus externus. These sort of spike a little bit more frequently. And the D1 positive receptors in the direct pathway actually a little bit more hyperpolarized. They're less likely to spike. And so the idea is that when you put a tonic level of dopamine in, you're essentially balancing the activity of these two populations such that phasic bursts or phasic drops can shift you towards a D2 positive or a D1 positive regime that can either facilitate or suppress a movement. And so if we were to draw that out in cartoon form, we could imagine our little direct pathway striatal neurons here projecting down to the globus pallidus and then out to the matrix thalamus. And then we have our indirect pathway projecting to the globus pallidus externus, which then inhibits the globus pallidus internus before hitting the matrix thalamus and out. And so here we've got D1 receptors and D2 receptors on these two populations. Now, importantly, D1 receptors are actually going to increase the excitation of the system using something called a G protein, and D2s are going to inhibit it. So you can actually think of D1 as exciting the system and D2 as quelling the system down. And so this has been known for some time, but one thing that I think is less well known is that different types of neurons in the cortex actually project to these two different pathways. So the ones that I've been mentioning today, these layer 5 B cells, these ones with these big fat stalks into layer 1, actually preferentially synapse onto D2, the D2 expressing indirect pathway neurons, and a completely different population called the layer 5 IT neurons. They have a much less extensive stalk up into layer 1. They actually are listening in a lot to layer 2 and 3. But without having elaborate trees. They're much more like an integrated fire neuron like this than this context-dependent firing neuron. They project into the D1 system. But really interestingly, these two populations also express the same dopaminergic receptor as the pathways to which they project. So that means that this cell here that's projecting into the direct pathway will respond to dopamine by increasing its firing. And this cell here will respond to dopamine by decreasing its firing in the same way that this would as well. So what that suggests to me is that the balance between these two different pathways is actually extended across away from the striatum into the cerebral cortex as well. And I think thinking then carefully about how the ventral tegmental area, which projects to the cortex and releases dopamine there, balances with the substantia nigra pars compacta, which projects to the striatum, is actually a really, really important set of mechanisms to understand in order to understand the impact that we're giving someone when we present dopamine like a precursor like L-DOPA or a dopamine agonist or antagonist to the system. So if we just imagine a projection now from the VTA and the SNC heading into this system, what that means is that you could imagine some, let's say, arbitration between the different populations. Let's say that there's a population coding for a purple action versus an orange action. They kind of fight it out in this kind of competitive model in the D1 system. And whichever one wins, let's say that it's the purple population, it then can inhibit the globus pallidus population that represents that movement. It releases the matrix thalamus. That then allows you to transition from an information accumulation mode where you're trying to vote for the two different options to essentially hit the apical dendrites of these layer 5 neurons, which is then going to allow them to transition into a burst mode. They will then hit the indirect pathway, shut off the basal ganglia by telling the globus pallidus it's time to work again, and project down into the brainstem to the pontine nuclei, hit the cerebellum, and enact the movement. And I think this is a really, really interesting way to think about the system, because it's rather than being striatally focused, it's now incorporating the benefits of having the thalamus and the cerebral cortex and all of their computational capacity. It's baking them in to understand the system in more detail. So I think this is a really fascinating idea. It turns out that that other people have had similar ideas long before I did. This happens a lot in science when you go back and read the literature. So there was a really lovely paper that I just want to highlight from Baraselli and colleagues, where they corral a lot of the more recent evidence and shift the kind of thinking from this traditional go, no go type pathway where the direct pathway controls go and the indirect pathway controls stop, to this more competitive idea of approach and avoid, where you've got different populations of direct and indirect pathway neurons essentially voting for their best option, and whichever one wins ends up being the one that captures the action. So if you're interested, you could read a bit more in that work there. I also think this helps to explain a really fascinating and rich literature in birdsong. And I just have to highlight this because I think it's an absolutely fascinating result that I don't think gets thought about enough. When we think about basal ganglia and dopamine, we often think about habits and well-formed behaviors, but we discount the fact that the basal ganglia can actually act on different elements of a behavior at different points in learning. And the birdsong literature is an absolutely fascinating example of this. Although the basal ganglia does not look identical to the one that we see in humans and monkeys and rodents, it's a little different. There's some weird different synapses in there. The basic architecture is very similar. And what happens in the case of birdsong is that this little bird here, when it's born, has to learn how to sing if it wants to end up attracting a mate. And the way it does that is it learns by listening to its father's song. And its father sings a particular song, and it does its absolute best to mimic that song. Its job is to make the song as beautiful as it can. But just to match that father. When it becomes an adult, the last thing he wants to do is sound like his father. So he tries to riff on that original song and make it his own, do something different with that song and make a change to it. It turns out that if you lesion the basal ganglia early in the life of this bird, it never learns its father's song. And if you lesion it late, it has no impact on its ability to sing its father's song, but it can't riff on its father's song. So in other words, the basal ganglia is important for the accrual of information early on in the course of this animal's life, but in the elaboration of the information later in life. And I personally think that thinking about the system as a whole and incorporating the complexity of the thalamus and the cortex can essentially provide us with rich explanations for how that might work. So I've tried today to kind of walk you through a lot of quite complex material, going from the anatomy of the thalamus down to the cellular level, trying to think about how we can get intuitions for how that might work through models, taking seriously this notion of different types of information processing via the subcortex, and then thinking a little bit about how the neuromodulation of this circuit might work at that kind of more zoomed out circuit level. I just really wanted to quickly bring this back to Parkinson's So we've known for a long time that the thalamus is a really key target of abnormalities in Parkinson's, but I don't think we've ever really had a great place to put it. And so, there's really good links between things like depression, cognitive impairment, gait freezing, and hallucinations that each map to different parts of the thalamic architecture. And I really want to motivate people to be excited about this opportunity now to start to bring the thalamus and the cortex back into the story about how different Parkinsonian symptoms, particularly these more paroxysmal symptoms, might be conducting themselves down at the cellular level. So to give you a kind of little take-home message, perhaps we could think of the pervasive symptoms as inhibiting the brainstem structures that control gait, but then these more paroxysmal symptoms, it might be related to this more flexible pattern of activity that we see that is mediated by the diffusely projecting thalamus. And with that, I'll say thank you very much and more than happy to take any questions.
Okay. All right. Well, that was great. Thanks very much. Actually, just a comment. I think, one thing you didn't mention as a paroxysmal symptom, which might fit into thalamic function, would be cognitive fluctuation.
Yeah.
Which are such a prominent feature of more advanced PD. And again, if
Yeah.
you're thinking about the thalamus as sort of gating these type of recurrent phenomenon, then that would be, I think, a natural one. Because sometimes in some patients, the levels of alertness and changes that we see are just so dramatic with some of these individuals.
Yeah, so I've thought about that a little bit with a colleague, Eli Matar, who's now finally back from his fellowship at Queen Square. And so we've got that in our crosshairs as something to talk about next. And one of the problems with reading the literature in the neuroanatomy literature is that it's sort of endless. And there's been similar inroads to understanding the complexity of the arousal system and how it's distributed across the brain that I think are another really great place to think about some of those fluctuations. And so, yes, I totally agree with you, and really fun to start thinking about this, but hopefully bring it out to more.
Yeah, so I think Tom Wickman, you have a question? So Tom is deeply interested in the thalamo and the thalamus and the cortex. Go ahead.
Yeah.
Tom,
Yeah.
I think you're, there you go. Okay.
Yeah, no, thank you. But I think Steven had his hand up first. Maybe he should ask for.
Okay.
First.
Dr. Shine, excellent lecture. I really enjoyed it. My question is, do you believe that these diffuse matrix type thalamic projections to the extragranular layers of the cortex, that they are the organizer of self-organized criticality in the cortex? Is that kind of what you're insinuating?
Depends on those terms can be a little loaded. So I think we need to be a bit cautious with the term organizer. There's a lot baked into that. I personally think that dynamical systems language is the most profitable way to think about the nervous system. And when you take that seriously, you're faced with a set of challenges. One is asking, what type of dynamics is this system instantiating? And that's one question. Another question is, what kinds of order parameters could I calculate from the system? So how can I tell whether or not the system is currently functioning as it should be or not? And different circuits have different types of order parameters. And then a third thing you can ask is, what's a control parameter for that system? What type of parameter is changing that system so that it has this type of activity versus that? And I personally think that those kinds of control parameters are all throughout the nervous system. I think the ascending arousal system is most probably a great place to start looking, but I think that neurobiology has found a way to mimic these control parameters in glutamatergic circuits as well. And this is one of the kind of complexities of brain evolution, right? Is that if there's a good trick and it's pretty cheap to create, brains will have figured out ways to do it over time. And so looking back and trying to find like a culprit for the organizer or the control parameter, I think is probably not going to be the most profitable way to think about it, but rather, is this circuit that I'm looking at currently one that is instantiating the order or acting as a control parameter for the order? And if you take that lens, you can then decide, well, should I perturb that system in order to change this one? Or should I just ignore this one or track this one, etc.? So that's a more profitable way to think about it.
I see. Thank you so much.
All right, I think, okay,
No worries.
Tom, I think you're next.
Yeah, right. So I thank you for the really great lecture that you gave there. whirlwind tours through the whole system. So it's very, very cool. I was wondering whether you had incorporated in some of these models some of the secondary anatomical changes that occur in relation to Parkinson's. So the loss of centromedian nucleus neurons, corticostriatal plasticity, corticosubthalamic plasticity, or the loss of dendritic spines at the cortical level. So all of this will influence processing in the Parkinsonian state. So has that found its way into these models yet, or is it still to be had?
Yeah, so absolutely not at all, and would be something that I would very much love to learn more about if there are sort of syntheses of that space that could be brought to bear on these models. Yeah, it's been, as I mentioned, it's been a while since I was really close to the Parkinson's literature. I think I got to the end of my PhD and had a lot of, I thought, really interesting ideas about how the system might function or dysfunction, but really no tools to be able to actually answer whether those questions were just cool stories or actually defensible. And so a lot of the time that I've been spending, in the last 10 years or so has been trying to advance the tools and skills so that I could come back and ask those kinds of questions. And so, if you have a set of, a synthesis of those, or if you'd be ever willing to chat, I'd love to learn what the current state of the field is in terms of those structural abnormalities, because a lot of those areas are super important. I think you're right.
Yeah,
Well, so
I'll do that
yeah,
too. Reach
Okay,
out.
great.
yeah,
I'll reach
Tom and
out.
his colleagues have done a lot of interesting work along these lines, actually. So
All right,
very
I'd love to learn
interesting.
more. Yeah, great.
Yeah. All right, Nico.
I have two questions for you. Thank you for, in the first place, Mac. Excellent lecture. I really enjoyed that. My first question is here in the Udal Center, at least at the Human Research in Parkinson's, we have become very interested in upregulation or compensation of cholinergic nerve terminals. What do you think, based on the specific projection neurons and connectivities, do you think maybe underlying such an imaging observation that we believe may be compensation?
Yeah, that's a really great question. So we've been doing a bit of work on the cholinergic system in the healthy brain, and one of the really fascinating things about the cholinergic system is that it's quite different than the dopaminergic projections and the noradrenergic projections and the serotonergic projections, and it's quite targeted to particular areas and quite selective, so that it projects in ways that, let's say, rather than kind of diffusely blanketing an area, it comes up and hits this area, but definitely nowhere around it, and then it goes and hits another area. And so we've been thinking a lot about it as kind of a really great way to stabilize those areas and let them become active as a coherent distributed network, as opposed to just open up the possibility that anything could become active. And so one way of thinking about the cholinergic system becoming upregulated is that in the Parkinsonian brain, you essentially know that you can't use dopamine, that's gone, and you're like, what else do I do? And you turn up the gain on these other systems, but maybe that ends up essentially trapping you in this kind of more stable cholinergic state, and now you're not inflexible, and you essentially just have to act the way you act. That's very speculative. I don't have any evidence for it, but we could start to ask some of those questions, hopefully with the tools that we're developing on brain imaging and modeling. We could start to make some inroads there. But yeah.
Okay. Yeah, excellent. Yeah, my second question is a little bit generic and perhaps a little bit theoretical, but several times you mentioned the connections and connectivity with the cerebellum. From the individual long axon neuroanatomical research studies that you're doing, have you learned anything about what for me remains a little bit elusive, the cerebellar locomotor area, which supposedly is in a white matter close mediolaterally to the vermis, but not in the gray matter? Have you found anything about connectivity in the area that the imagers of the imaging community believes is where the cerebellar locomotor area is?
No, the short answer, Nico, is that I have not seen anything really great along those lines, but I'll put it on my list. I'll go have a click and I'll let you know if I find anything cool.
Okay, yeah, thank you.
All right, very good. Okay. Well, let's thank Mac for an excellent talk, and particularly early in the morning, much more cheerful than I would be in the early morning. Thank you so much. That was really that was a tour de force tour, actually, really great. So thanks so much, and
Thanks very much for having me.
all
Thank you.
right, great.
Okay.
Thanks to everybody.
Bye-bye.
Run.